Today, I'd like to introduce our speaker who will be talking on establishing reference data and call sets for benchmarking cancer mutation detection using whole genome sequencing. Dr. Litai Fang is currently a staff scientist at Endpoint Health, a company dedicated to finding targeted therapies for critically ill patients. He worked on the STQC2 somatic reference project when he was at Roche Sequencing. Previously at BINA, he led BINA's team participation in the ICGC TCGA DREAM Somatic Mutation Calling Challenge, where his group finished #1 and #2 in the stage 5 indel and SNV sub-challenges. And he's also developed software for detecting somatic variants called SomaticSeq. So Dr. Fang, please take it away. Dr. Fang, you may be on mute. We can't hear you.
Thank you. Yeah, all right, sorry. So diving right into it. So I'm going to talk about the consortium's effort to establish a reference sample for the benchmarking of somatic mutation detection technologies and algorithms. So we chose a tumor normal cell lines of triple negative breast cancer, where if you can look at the karyotype for those two cell lines, on the left is the tumor cell line. And there are actually, if you count, well, I didn't count, but if you count, there are 66 chromosomes, and you can see, a ton of rearrangements and copy number variations just by looking at the karyotype, and no one can identify what the chromosomes are. On the right side is the chromosomes for the normal cell line, and where the chromosomes look more or less normal, except for, one maybe rearrangement between chromosome 16 and chromosome 6. But overall, the normal cell line looks quite normal, where the tumor cell lines look extremely rearranged. But I will mostly focus on the small somatic variant results, SNVs and indels. So the results, if you look at their variant allele frequencies for the somatic SNVs, single nucleus substitutions on top, and indels on the bottom, especially on the top. If you look at somatic SNVs, you can see that the variant allele frequencies ranges pretty much between about 2 to 3% on the lower end and 100% homozygous variants on the upper end. Not only that, you can notice that there are certain peaks at about 66%, 50%, 33%. And those actually represent variants that occur in regions where different copy number states are. If you look at the indel, though, you don't see clear peaks, and you may even notice that the variant allele frequencies for the indels seem to be lower overall than SNVs. And that may be due to the fact that usually when you try to estimate a indel variant allele frequency, you tend to underestimate, because for short reads, which is the technologies that we use here mostly, if an indel occurs at the end of the short reads, it tend to be soft clipped alignment instead of an indel alignment. So they tend not to be counted when you simply count for indel reads. So that's the reason that the indel estimates seem to be a little bit lower, and that's usually the case. And if you look at the variant allele frequencies for the somatic variants across the tumor genome, which is we're showing here, chromosome 1 here, chromosome 2 here, and the variant allele frequencies are the green dots that goes across the genome. And if you see different colors in the genome, those are estimated copy number changes based on a software called ASCAT. So I'm not going to focus too much on the accuracy of the copy number estimates here. But you can see that when the color changes, then the variant allele frequency shifts. So they are tightly related, right? So so green, for instance, green panel means you have a variant allele frequency between 100% and 50%. So represents either a homozygous mutation or heterozygous mutation. And if you see lower variant allele frequencies, they represent subclonal mutations. And then when the copy number shifts to 3, then you see, 100%, though that probably means 3 out of 3 copies. 66% here, 2 out of 3 copies, 1 out of 3 copy here, and then subclonal. So between the variant allele frequencies and the copy numbers, overall, they are consistent with each other. And if you look at the variant allele frequencies at regions of the genome with different copy number states, and then it's clearer that, where the copy number is 1, you have mostly mutations with about 100% allele frequency estimates. At copy numbers of 2, you have peaks at 2 out of 2, 1 out of 2. At copy numbers of 3, you have peaks at 3 out of 3, 2 out of 3, 1 out of 3, and same thing for copy numbers 4 and 5 and 6. And then, but you always have subclonal allele frequencies where the mutation occurs only in a subset of the cell samples. Then I'm going to here talk about how we establish the reference call set. It's a set of somatic mutations that we are very confident they're real in a high confidence regions. And not only that, inside the high confidence regions, which represent about 82% of the genome, there are very few, if any, mutations that we have missed. And the reason is because we use a total of 22 sequencing replicates from 7 different sequencing centers with different aligners, different colors, and 2 separate machine learning algorithms to classify the mutations. So we start with a total of 21 whole genome sequencing replicates from 7 sequencing centers with the coverage somewhere between 50 to 100x each of them. Right? So they are, we have 3 high-seq sequencing replicates from Illumina. We have 9 NovaSeq replicates also from Illumina, 3 more replicates from Fudan University, 3 replicates from Novartis, and then 1 of each replicate from ITERIS, NCI, and Loma Linda University. And then we have grouped them into 5 groups. The first 4, basically, they came from the same sequencing center. So if there are any specific errors or biases relating to each sequencing center, they are grouped together, and then we can look at if you have variant calls that only comes from one center. And then for each of those sequencing replicates, we align them with 3 different aligners, BWA-MEN, both type 2 and Novo Align, and to create here a total of 63 tumor normal BEM files, 63 tumor normal alignment maps. And then we use 6 different somatic mutation callers to discover mutations in each of the 63 tumor normal BEM pairs: MUTEC2, somatic sniper, Vardic, Muse, Stroga2, and TN-scope, which is a caller from Sentient. Then for each of the 63 tumor normal pairs, we use 2 different machine learning algorithms. One is somatic-seq, the other one is mu-somatic, in order to classify mutations here for each of the 63 data sets. Right? So what we do then is to integrate the 63 calls, 63 classifications from machine learning algorithms, and to see how reproducible they are across aligners and across sequencing centers, and to give them confidence labels based on their reproducibility. Notice that for the 21 replicas that we started with, their depth is between 50 to 100x. But in order to rescue some variant calls that may have very low frequencies, low frequency means that they're not going to appear in all of the sequencing centers or all of the replicates, but they are going to be occurring only in some of them. And in order to increase their confidence, in order for us to know whether they really are mutations there or they just appear a few times out of random chance or a few times because of sequencing errors, we use an additional data set. It's about 350x coverage from Genentech. And then we also combine 9 NovaSeq replicates here, and then to rescue, potentially low variant, low VAF, low frequency variants, and that was deemed low confidence because they only occurred a few times in all of the replicates. And then we also combine all of them once to a total coverage about 1,500x, and to rescue a few more variants. And finally, we also cross-reference the variant calls with PacBio long read, and we will remove variant from the reference call set if they're deemed to be in conflict or inconsistent with PacBio alignment. And based on the reproducibility across sequencing centers, sequencing replicates, alignment, different algorithms, as well as PacBio, for the variants that we deem high confidence calls, we label them really true semantic variants. And for the variants that we are not totally sure if they're real or not, we remove their positions. We remove the region that contain those low confidence calls from the high confidence region, so that within the high confidence region, we are quite confident that the calls in the reference call set are real mutations, and there is very few or maybe no real mutations other than the variants that we have put into the reference call set. So two critical methods, right, in determining the reference call set are the two machine learning classifiers that we have incorporated into the workflow. So I'm going to talk a little bit about how we build the models that classify mutations in each of the 63 data sets. Because we have multiple replicates for many of the sequencing centers, like from Illumina, Fudan, and Novartis, we can take two replicates. And you know that, right? We know if we run the semantic workflow with two normal replicates, we're going to discover semantic mutations. We also know that those are not semantic mutations really, because they are actually the same sample. They're both normal samples. So any semantic mutations that you do call are false positives. So we can label those variant calls as false positives. And then we spike mutation into one of the replicate, and then we call that a synthetic tumor. So how we spike in mutation is that, we randomly pick a position. Let's say we're going to make a semantic mutation in this genomic position. So we will randomly pick some reads, and then we will change their base, right? For example, from G to a C, and then here also from G to a C. And then once we change the read in those reads, we will align them again, just to make sure that you will have the same aligner scores if the reads are those variant reads. And then those mutations are spiked in at different variant allele frequencies to ensure that our machine learning model will see a range of different mutation frequencies and different positions. So that's how we produce a synthetic normal for machine learning labeling. Then we will just run that, run a tumor normal workflow with the same 6 mutation colors that we have used. Then for the mutations that we have spiked in, and if they're discovered, most of them are, then they will be labeled as mutations, right? They will be labeled as 1. And then everything else that we didn't spike in is labeled as a false positive, because we know that those are not semantic mutations. those are two normal replicates where the only mutations are the ones with spike. Then based on each of their genomic and sequencing features, so for each variant calls, each mutation calls from this data set, we will extract using semantic seek things like, what's the color classification? Did Mutek call it? Did Muse call it? Did Varda call it? That's a feature. What's the mapping metrics, right? What's the number of misalignment within 10 bases, for instance? What is the mapping quality? Is it a DB SNP variant? Does it have strand bias? What's the base score quality? And for a total of 100 different features for each variant calls. And then we will label each of them as true positive or false positive. Then all those features and the labels will be fed into a classifier, two classifier here. One is a semantic seek machine learning algorithm. The other one is new semantic. And for semantic seek, we actually create a different model for every sequencing center and every aligner in a way so it will learn, right? It will learn the sequencing errors and maybe the sequencing biases from every single sequencing center, and then we will apply that to the data that came out of that same sequencing center. For new semantics, we actually combine all of the training data into a single classifier, well, actually one single classifier for each aligner, and that has SNVs and indels both in it. So we actually use three different new semantic classifiers, one for each aligner. And Mohammed will talk more in detail about new semantic in about a month. So we have the classifiers, and we can use it to classify the mutation calls that comes out of each sequencing center. So based on the reproducibility for each of the mutation calls, we classify them into 4 different confidence levels. High confidence and mid confidence are eventually what we consider as true mutations, and they're included in the reference call set, whereas low confidence calls and unclassified calls are not included in the reference call set. And here we can, maybe you can see why. Out of 63 data sets, right, 63 tumor normal band files, for the high confidence calls, for the high conf calls, on average, out of 63 tumor normal data sets, you have over 60 passes. So that's over 90%. For those mutation calls, over 90% of tumor normal data sets, tumor normal band files, they're classified as a real mutation. And then 0.15 reject means that's how frequently they are classified as a false positive by semantic seek. So very rare. And you have rates. So that's like a 200-to-1 ratio. So they're highly confident. For median confidence, so for mid confidence, it's slightly more challenging. On average, you have fewer than half of the samples where they are classified as a semantic mutation by semantic seek. And on average, there's almost one sample for each where they're classified as a reject. So there is a little bit of conflict there, but not too much, right? The ratio, if you look at the ratio, is like 50-to-1. And they are mostly low VAF variants that's not captured in all of the replicates. They're only capturing some because the VAF is low, and that is not unexpected if a variant is low VAF. For low confidence mutation, it's more challenging, and I would say impossible to resolve. Their allele frequency tend to be around 2%, 3%, or even lower. And on average, if you look at the semantic seek classifications, on average, it's about 8 pass samples versus 2 reject. So here the ratio is about 4-to-1. You still have, right? You still have more pass, more semantic mutation classifications than false positive mutations, but it's not overwhelmingly majority. So those we cannot confidently distinguish from noise, and we are not certain if they are true positives or false positives. So they're not included in the reference call set. And for unclassified calls, here you have more reject calls than pass calls. So they are mostly classified as false positives, and they mostly occur in regions of challenging regions, uncallable regions. Some of them have very low mapping qualities, and sometimes they're clearly germline variants if you look at enough samples. And many of them are not really SMVs or indels. They may be real variants, but they're part of complex variants that's out of scope for SMVs and indels. So we'll show you some examples here. So for the all the calls that we have classified, so that's the superset, all-inclusive set. The most of them are high confidence, right? So some variants are very easy to detect across the samples, across the sequencing centers, and that's the number here. And then a lot of the other variants, they're just you have mutations you have detectors that call them, but they're clearly false positives if you're looking deep into it. The ones in the middles are a little challenging. For the metconf, we're still pretty confident. We're still very confident that they're real mutations, so we included them in the reference call set. Where for the low conf, we're not sure, so we actually just remove them from the high confidence regions. And right, that remains for the future if we want to reclassify them one way or another. And semantic seq classification is consistent across replicates and aligners. And that is despite the fact that the classifier was built with different models. So the different model was built for each sequencing center and each aligners. So in that way, that reduces overfitting from each one data set, but they're still highly consistent across all of the data sets, right? So on the green arrow shows you the direction of the plane where the number of samples a variant is called a pass. A pass means a true mutation classification. So for a variant that's called a pass, over and over, many times it will be in that direction, right? The green arrow. For a variant that's labeled a reject, and that will be in that direction. And if you have variants, if you see data dots in the middle, that means there is a great deal of conflict between between different sequencing centers or liners for semantic seq. You almost don't see them here for SMVs, right? All of them are either there. So that means in this corner means all of them are labeled pass, and very few of them are named reject. And the black ones are the unclassified variants. That's likely false positives. And you see a high number of reject, but not many passes. And the z-axis is the variant allele frequencies, and you can see that for the unclassified variants, the VAF is pretty much across the board from 0 to all the way to 100%. So they're not rejected because there is not enough signal for them, but because there is bad mapping quality, bad alignment metrics, and things like that have them labeled as false positives. Now there's a little bit more conflict for indel. You can see that, right? Overall, the shape looks the same, but there is a cluster of variants in the middle here. That means there's conflict. So if we look into it, many of them have about 30 to 40 pass classifications and somewhere between 10 to 20 rejects. That's a great deal of conflict between different sequencing alignments and centers. And so we actually look into it and why that is. And the reason is that many of those indels are pretty long in the sense that they are deletions of bases around 10, right? Somewhere between 8 to 20, 15 up to 20. And since they are pretty long indels, they're really at the edge of detection for short resequencer and typical indel workflow. And it's hard to detect them because they pretty much have to be in the middle of the read to be aligned at indel. If they're toward the edge, it will be a soft clip reads. So they tend not to get detected in some of the aligners. But when we look into the alignment, they are clearly indels. And so they were originally labeled as medconf, but we actually look into all of them, and it's pretty clear that they really are mutations, and so we classify them as high conf here. We also looked at to see if somatic seq and new somatic seq and new somatic classifications are consistent, and they're highly consistent. So on this 3D graph here, on this axis, that's the number of times a somatic seq has labeled the variant as pass. And on this axis, it's new somatic. And it's almost always on a diagonal. And if you look at their Pearson's R, it's almost 1, it's 0.997. That means a variant, if it's classified certain way by somatic seq, it's almost certainly classified the same way by new somatic, even though those are two totally different algorithms and actually use slightly different training data sets. Somatic seq is trained on each sequencing center, whereas new sematics is trained with all of the sequencing center combined. And also, if you look at the histogram to see how many times a mutation has been classified as pass by either of those two either of those two classifiers, that gives you kind of a confidence that most of the variants in the data set, in the call set, they're either all of them call them a mutation here, right here 60. So that means every data set it's classified a mutation. And then on the left side, you have very few data set called a mutation. So you just looking at the graph, you can say, right, exclude those variants, include those variants. It's ones in the middle that's challenging. We also did orthogonal validations for our call set. So across the different confidence labels. Upper left is done with AmpliSeq for coverage about 2000. Upper right, that's the VF scatter plot for ion torrent whole exome sequencing at a depth about 34x. Lower left is whole exome sequencing, but with HiSeq. So it's sort of the same platform, not exactly orthogonal, but still it's different library and a different sequencing setting. Lower right is PacBio. So one thing you notice is that if you see solid dots here, that means a variant is validated on this platform. Validated means here really means there are enough reads in the variant. There are enough variant reads in the tumor, and there is no variant read in the normal. And if that threshold is met, it's deemed as validated. A variant is not validated. That's an open circle. It means that right they meet those threshold. So right, all of the green dots here are solid, are validated, almost all of them. And if you see data points on the bottom, then the x-axis is the variant allele frequency estimate from the whole genome sequencing that we use to classify the reference call set. And on the y-axis is a validation data set, right? So if you see things on the bottom, it means there are variants that's classified as high confidence, and those are their varying allele frequencies, but you don't see them in here in AmpliSeq. And then here, right, it's like there's some of them you don't see being ion torrent. And so all the green things are in the middle, for things outside, they're mostly black, a little bit of orange. Orange is low confidence. So that, and then some of those so-called not validated, they're actually false negatives in the platform. But overall, you can have pretty good confidence that our high conf and mid conf variants have extremely high validation rates, at well over 99% for SNVs. Indel slightly lower. There are two indels that weren't validated in in the validation platform. And for caution, we just remove them. We actually remove those two variants, two indels from the validation, from the reference call set. And we also looked at some, low confidence unclassified calls that are seemingly validated in the validation platforms and want to see what's going on. And most of them belong to two classes, right? Why are they validated? One is complex variants. So on the left, this is a tumor alignment. This is also a tumor alignment. Here there is a variant here. It was not classified as a somatic mutation by somatic seq or new somatic frequently, because it is right next to a 13 base deletion. Here on the normal, there is nothing there. Everything is reference. And that is a unclassified call. It is not a reference somatic mutation call. Now, this may well be a somatic variant, but it's a complex variant. It is not simply an SNV. And so that may represent some shortcomings for our reference call sets, that we really identify SNVs and indels. And for complex variants, we're mostly not considered them. On the right side, it tells you the advantages of using multiple alignment. On top, there is, even though you can see, right, there's a sudden coverage drop, and the SNV alignment is within this seemingly deletion, but there are some reads that BWA has aligned it as a variant read here, and it's being classified as a somatic mutation at least once. That's why it's there in some samples. And here's the one in the middle is novel aligned. You don't see that variant alignment here. And on the bottom is the normal, and it looks totally normal. Everything's referenced. The coverage is same across the board. And so it's, quote unquote, validated in the validation platform simply because there are enough variant alignment to meet a certain threshold. But this is also pretty clear that it really is a false positive, even though it's quote unquote validated. It's not a real mutation. I'm also showing, the power of using data sets from multiple sequencing centers at a total of 1500x, and that gives you information that you will never get from a single sequencing data set. So as an example of using our reference call set and high confidence region as a benchmarking tool, we ran StrokeGut 2, one of the data set that we use here. It has about 100x in depth. So after we ran the StrokeGut 2 results with our reference call set and the high confidence interval, it has a sensitivity. StrokeGut 2 give you a sensitivity about 92% and a precision about 87%. And so that's very good. Now, the interesting thing here, I guess, is that even though in our reference calls, right, remember most of the calls in the reference call set are labeled as high confidence. Only about 5% of them are labeled as medium confidence. Medium confidence because they're not reproduced across all of the sequencing centers or aligners, but only some of the sequencing centers due to the fact that they are low VAF. Right here, in the reference call set, only 5% of them are medium confidence. But here, the false negative, right, the mutation that StrokeGut 2 did not detect in this data set, 60% of those missing calls are medium confidence variants. Now you're wondering, is it because those are actually false positive in the reference call set, or they're false negatives in this particular data set? And so after examining what those calls are, I can say with confidence that they are false negative in this particular data set. And here is an example, right? So on top, we're actually looking at the combined alignment from a 350x data set. On the bottom is the normal. So you can see that if we look at a high depth, if we look at a 350x data coverage, there are 9 variant reads in it. And because we have used 21 sequencing replicates plus this data set, we have put this variant into the reference call set. But in this particular data set, this replicate, even though it's 100x, there's only one variant read. So there was just not enough signal to call that mutation in any single data set. But since we have all those data sets put together, we can, in a way, confidently call that mutation a real somatic mutations. And on the flip side, there are germline variants that we see with all those sequencing replicates and also combined data sets. But sometimes in one data set, you don't see them simply due to sampling errors. Here we can confidently call that a germline variant, or in any one particular data set, you will not be able to do it, right? It's not Strogat's fault, but simply because you can't see all the signal in one particular data set. And the data are already deposited in SRA, and the call set is in NCBI, right? Everything is open and you can download them pretty easily. The paper describing this work is now accepted. And the documentation is also detailed every step of how we come up with our reference call set and high confidence intervals. And acknowledging over 200 members and 60 institutions that involved in this work. Now, time for questions.
Litai, thank you very much. That was very informative, very interesting work. And I'm going to give the people some time to submit questions. Let me just ask, I guess, a question of my own first, and then I'll get to everybody else's question. So you had mentioned, through the first few rounds of the analysis, you were using data from multiple groups, multiple variant callers, things like that. And then later on, you used long reads like from a PacBio to help with the validation effort. So I guess I just wanted to find out from you, how important was it to follow up with, say, other methods? I know you use also Thermo Fisher and AmpliSeq and things like that, but can you speak to like the long reads of PacBio? How useful was that in terms of helping to clarify these putative variants in terms of high and low confidence?
Yeah, so they are important. We did go through a few iterations. The high confidence calls, right? For the calls that's called in every sample, that's simple. And for the calls that's called in only a couple of samples, that's also simple. Those are either true positives or false positives. But there are calls that's somewhere in the middle. So after the first round of validation with AmpliSeq and Ion Torrent, we went back and retweaked the threshold a little bit to once we understood that the fact is that if a variant is classified as true positive in a number of sequencing centers and the number of aligners, but if they're not discovering other sequencing centers or other replicas, that's okay. They are still likely true positives. But if they're classified as like reject, then those are likely false positive. What I mean is that if you have a variant that's called in, let's say, 10 data sets out of 63, if they're low VAF, then they're actually likely to be correct. If they're high VAF, they're likely due to like false positive markers. So those are the things we learn as we look at the validation data and figure out like what's actually variance and what's false positive.
Okay, thank you. And so we do have a question from Ken Gang from Thermo Fisher. He was commenting on the large number of unclassified calls, and compared to say the high confident group, that it seems like there's maybe even more unclassified versus high confident. But those unclassified calls had to come from someplace. Yes. In other words, they had to come from different callers, whatever. Any comment about aligners or sequencing depth, or maybe, what would be the reason why we would get so many unclassified variants, which means that, I guess in my mind, someplace, somewhere, someone called this as a potential variant. And it could be that maybe only one or two combinations did, as opposed to, your requirement that, the vast majority of systems, call a variant for it to be high confidence. So can you just classify on the reasons for why there are so many unclassified calls? And should we be scared about that, or is there a reasonable explanation, things like that?
Yeah, so most of the unclassified calls, they are right. They're likely false positive. And most of them are due to they're called false positive. And when I look into sequencing alignment, mostly due to likely alignment errors. Is that if you look at, there is a SMV alignment here, and then some caller has called it, and then there are like, you know, three different mismatches right next to it, but you don't see those mismatches in a normal. So, right, so they could be mapping errors and likely to like mapping errors, or there could be some, right, or it could be there are actually multiple somatic variants, somatic changes successively. And that's sort of our limitation. We tend to think them as false positives because multiple somatic changes within a short distance is highly unlikely. And I wouldn't say, be scared of them, because most of them are pretty clear. They have clear markers to indicate that they're false positives, and you can filter them out if you look for it. You do run the risk of missing actual variants of those characteristics, but vast majority of them, they are actually false positives and not real mutations.
Yeah, so complex variants like that somatically seem to be rare, and therefore you're fairly confident that they were, misalignments or other things like that. I mean, that's certainly a lot of methods that do even calling, try to take that into account. And I think, for example, having the normal, there as well helps out, like you said, with that, especially with your, machine learning methods. Okay, so I have a question from Naomi Thompson about the next project. Are there plans to tackle the challenges of more complex variants? And maybe this will also give you a chance to speak about what you had talked to me about earlier, which is a kind of a related question, which I'll follow up with a little bit later. But a related question is, is there a follow-up even to this study, for example, related to understanding how stable the assessments of these two cell lines are over time? So yeah, what about the next project? Either looking at more complex variants or looking at, follow-ups looking at stability over time. Can you speak to that?
Yeah, so complex variants are actually quite hard, and so I have to say there is no immediate plan to classify all of the complex variants across the whole genome. You can always do that for a subset, where you sequence it with different platforms and visually inspect them, and then do PCR. Over the whole genome, complex variants are hard to it's hard to kind of catalog all of the complex variants across the whole genome, because vast majority of them are errors due to mapping, and we rely on mapping, we rely on short reads for all those things. So if the vast majority of them are simply not true and there's often no way to distinguish, then it's kind of at the edge of what we can do on a whole genome scale. Over a short region, you can always throw more technologies at it, throw more data at it. On the whole genome scale, that would be very expensive. And the other questions in terms of stability of the cell line is the consortium do have plans to do the same study and look at the genomic changes as the cell lines, as the cell line, grow from one passage to another, and in order to figure out what are the genomic differences if you have like two batches. So that is in the plan, and so that we will do. Complex variants, we may have to we may either have to wait for a better technology that can be both long and accurate and cheap, or we may have to do it with a subset of regions where those variants are frequent and important.
Okay. We also have a question from Gunjan Hariani that asks whether or not these two reference materials are, or at least maybe the tumor one, because it conceivably you could get a normal from blood or whatever that may not have to be an FFP, but specifically wondering whether or not the tumor cell line is available in an FFP format that might be comparable to a sample matrix that individuals or labs or institutions would have when they have to study, because a lot of times tumors are preserved in formalin. So do you know anything about, I assume like for your study, these were all basically intact, fairly well preserved DNA, but I didn't know if you knew anything about whether or not there are versions of this that are stored in formalin, so you could understand what the impacts of formalin are, but still, use it to test methods that are, extracting nucleic acids from formalin, and then also testing whether or not they're sensitive and specific for these variants.
Yeah, so the purpose for the reference sample is to look for accuracies for different platforms and different library preps, including things like FFPE. So the consortium has actually done pretty extensive study on the effect of FFPE on the variant callings. I think Wenming may have talked about it last week, and the paper should be, the paper is also, the preprints also, you can also find the preprint, and the paper is also accepted. So there we have done formulins where the fixation time ranges between 1 hour to 24 hours. We've done whole genome and whole exome on those FFPE samples and looked at what are the changes, what are the effect of different formulin fixation time on the effect of variant calling. So that is definitely an important thing to look at. And the consortium has done some comparison studies on it too.
Yeah, so maybe the question is, these were maybe formalin preservations done by the consortium, correct? In other words, they received the intact material, then put it in formalin. And I guess the question is whether you knew of some institution that has already put them in formalin for a period of time and is making them available. But it could be but at least in this case, the consortium was the one that stored the cell lines in formalin under different conditions. Is that correct?
That is correct. Yeah.
Okay.
And right.
Okay. Well, let me ask related to the spike-ins that you mentioned. So I assume that the spike-ins related to positives in the cell lines that you mentioned were essentially in silico. In other words, you didn't, as opposed to spiking in some kind of artificial nucleic acid that was specifically constructed. I just want to make sure I understood the nature of the spike-in. Was it an artificial construction, or were you like in silico modifying certain bases so that you would have, in other words, like in FASTQs or BAM files, that you would have these variants at specific allele frequencies, so you could look at the noise in the normal samples versus not. I just wanted to make sure I understood about the nature of the spike-ins.
Yes, it's exactly that. So it's in silico, changing the base in silico, and the reason so the reason it's in silico instead of like putting actual reads in is that we take a BAM file from one sequencing center, and that BAM file has all the biases and sequencing artifacts associated with this data set and this sequencing center, and we go in just change one thing, change one base from G to a T or something like that, and then we randomly pick which reads we do that. Once we pick the variant site and pick the variant allele frequency, then we just randomly choose which reads we will make that change. And then after you make the change, you will realign that read to make sure that you're going to have the same mapping qualities and alignments as if the read were real.
Gotcha. And so related to that, was this something that you did using your own tools, or did you use an outside tool to assist with this?
Yeah, that's BAM Surgeon.
Okay, okay. And the reason I ask is, you mentioned some of the factors that different aligners use to consider, or maybe even the neural net, or I shouldn't say neural net, the machine learning method, might use. I may have said neural net because of one of your diagrams made me think it was using a neural net, but
one of them using neural net. Yeah.
Okay, yeah, the new semantic, yeah. But I guess I didn't know whether or not, for example, they may be picking up on things like strand bias or something else. And I didn't know if you, if the tool was actually conscious of strand bias and was making sure that when variants were injected, that they were representative and therefore didn't have an association with strand bias. In other words, you actually stratified random versus just say random. But the thing is, if you did like perfectly random, it probably ended up being stratified in one sense, and so those strands were probably hit. But I guess I just didn't know the nature and how what the details were of the injection. But you used the tool to assist with that. Okay. And maybe the last question I had was related to using these reference samples in general. What do you view as how the general community will be able to use these reference samples? Who do you think will best take advantage or best help either institutions or companies or individuals? Who is this going to help the most, and how do you envision people using this knowledge?
Yeah, I think one of the things that is to build better algorithms that build better machine learning algorithms in order to tell what is actually a real mutation versus false positive. So Mohammed, in about a month, will actually talk about the new model that he has built with the SEQC2 reference data sets on the new semantic tools, right? So the models that he has built here is based on the same in silico spike that we have helped build this data set. But once we have the call set, reference call set, then you can test your semantic mutation callers or any algorithms and to see what you are calling correctly and what you are calling incorrectly. So, and the things downloading data is pretty simple. So that's something I can see that can be done pretty quickly. And then there are also some samples remaining where people can benchmark new technologies and things like that.
Yeah, exactly. So so whether you're just on the in silico side and you download this data and you test out new methods for detection, or whether or not you're bringing online a brand new assay. And I guess the nice thing is, whether your assay could be, it may not be whole genome, it may be more focused, or it could be whole genome, but because you are giving data about the whole genome, potentially it can be used for a wide variety of different types of assays and technologies as they bring them online and want to validate. So So I think it's I'm just gonna say I think it's a great contribution to the community as a whole, in terms of being able to evaluate their systems and make sure that they're operating the way they think they are.
Yeah, I would say this work is more suited for whole genome or at least whole exome study because we kind of have a lower limit variant detection here in the reference call set, about maybe 3 to 4%, because we didn't look at like 5,000 50, we didn't look like 50,000 x ctDNA or things like that. So it's more suited for a large scale whole genome data sets. I think a different working group 2 or 3 may have samples that's better suited for like very low allele frequency ctDNA kind of data sets, where here it's more for a whole genome context.
Yeah. Well, Leetai, thank you for that plug, because I was going to mention a little bit later on, like you mentioned, in a few weeks, the other working group that is more focused on, panels and focus somatic detection will be having their set of talks. And in one sense, to me, I see them as very complementary. Here you have samples and methods related to more broad scale whole genome, whole exome, and then we'll also cover maybe more focused panels in a few weeks. Well, anyway, thank you very much, Litai. That was very informative. We're certainly glad to hear your results from your working group in the SCQC2, and we'll look forward to hearing more in the future.
All right, thank you.
Thank you.